So what if, you've run an experiment and you're finding no difference? What basically, what if you have a null effect or no effect? And this is something that is reported in this particular study um, of low intensity care options to prevent self-injurious behavior. This is an article in the Journal for the American Medical Association. And if you look here, you see that there's no difference between their three groups: their care management group, their skills training group, and their usual care group. That there are about as many self-harm events in each group. Note that this is a large study, so there were almost 19,000 patients that were randomized, and almost that many were actually analyzed in these three groups. Very small difference. Now, one of the things that an editorial said is, and this is by Stanley and Dixon in the Journal for the American Medical Association, JAMA. He said, although the findings from the study do not support implementation of the programs as tested, it may be that the implementation strategy failed, rather than that the therapeutic approaches from which their interventions were derived failed. That's an important distinction, and that's one that we're going to keep playing with today. They noted that around 31% of those offered case management initially accepted the invitation, and approximately 17% stayed engaged over 9 months. Skills training, 39% accepted the invitation, and only 2% remained engaged over 9 months. Now, I think one way of looking at this is that these are not representative samples, and we're losing a fair number of the participants, right? And what is it about the ones that we're losing? Might there be something unusual about them? Or unusual about the ones that stay? Perhaps, the ones who are staying are, doing enough better that they're willing to do the kinds of things that were required in this particular study. Okay, so let's go back a little bit and talk about a number of things. One thing is that sometimes it's helpful to know that an intervention is ineffective. Remember, we talked about mastectomies earlier this semester, and that surgeons believed that radical mastectomies were much better than other treatments, when in fact the data suggested that these differences were very small, if any, in terms of 25-year survival rate. And if we're also thinking about some of the other impacts of getting a mastectomy, including some of the disfigurement associated with it, it might not even have been worth it, right? Even if there had been a significant difference. Sometimes, what we see is that an independent variable doesn't make a difference. And, that it may be we're thinking, we're not exactly sure what's going on. Our mood is not impacted based on cash available. It may not be or reading scores may not be impacted based on online reading games. We don't know what's going on here. Is it that there's no difference? Is it that there's some some poor design issue or some obscuring factor? And those are things that we might have to really go through the study and look at it in order to make some decisions. And maybe we don't even particularly know until we've looked at other kinds of studies and data. One of the things, though, that we do know is that we need to be very, very aware about between group variance and within group variance, and that in general, we want our within group variance to be relatively small and our between group variance to be much larger, so that we're able to see a difference. That can happen in a number of different ways, by making our measures more sensitive, for example, by getting more data, more participants in our study. So, we need to look at a variety of different things. How many drops of dye are there? Do we have to have in our water in order to actually see a difference? So, in terms of ceiling and floor effects, that may be another reason why we're not seeing a difference. It may be our questions are too easy and everyone's getting them right. Maybe our questions are so hard that everyone gets them wrong, in which case we really need to be thoughtful about designing our assessment. And that's something that we need to be checking on beforehand. Now, manipulation checks can detect weak manipulations. They can also detect ceiling effects and floor effects. So in this particular situation, we're using a manipulation check. This isn't our dependent variable, but we would expect that if our intervention was effective, that we'd also see changes in anxiety in these three groups, and we're not. In this case, we're seeing that our manipulation actually worked, and worked in the way that we thought it should. Now, finally, sometimes we have design confounds that actually work in reverse, and actually cause some null effects. So, for example, GRE prep may have a variety of different things that is going on that negatively impacts the how people are performing. And so we have to at least consider that as an option. How might that play out? And I'm going to stop here, and I'll see you in our next video. Take care. Bye.